Okay, we are going to be doing about extracellular matrix, which is basically about collagen and elastin, which are part of the fibrous protein. You all know that the extracellular matrix is the most abundant connective tissue. Okay, they say it's a well characterized fibrous proteins of the extracellular matrix, which serves as a functional structural integrity of the tissues, and it is a major component of all connective tissue, blood vessels, and also of the connective tissue of the eye, which is sclera and the cornea, both. Now this fibrous protein, which are forms the extracellular matrix, consists of basically E, C and M. M basically stands for matrix, okay, and E stands for elastin, C for collagen, and M for matrix. Matrix are made up of laminin, okay, then they are also made up of other proteins such as fibrin, fibronectin, laminin and proteoglycans. Fibrin, fibronectin, laminin and proteoglycans, okay, FFLP, that is what, and that is elastin and collagen. So these six basically become part of your extracellular matrix, okay. Now how are they different from globular proteins? Globular protein, you also know, are basically round shaped, whose shapes actually result because of different types of structure, like either being a secondary structure, tertiary structure, etc. Whereas these proteins, fibrous proteins, they exhibit mechanical properties due to its unique structure, which is obtained by combining specific amino acids into a regular secondary structural elements. Now, unlike the globular proteins, it's not like the same structure gets folded again and again and again. It gets folded and becomes a secondary structure, tertiary structure, etc. Whereas instead of that, here it is like there is a specific structure, okay, and this unique structure is because of regular arrangement of the connective tissue, regular arrangement of the protein structure. So it's secondary the structures of these proteins are due to the regular arrangement rather than the primary structure, secondary structure. Good. So the most abundant of all this protein we are going to see is your collagen. Okay, right, let me start inking. You all know that collagen is the most abundant protein in the human body. Okay, it's typically a long, they say it's a long rigid structure in which, three polypeptide chains are arranged. Three polypeptide chains, which are referred to as the alpha chains. Okay, so if you look at this picture here that is given here, okay, if you look at this picture here, you can clearly see that it is made up of three chains, right? So there are three chains here, okay, the 1, 2 and then 3. So three chains, less like your DNA having two chains, here it is three chains, and assume each of them is called as, say, a alpha helix, okay, referred to as alpha helix chains or alpha chains. So there are three alpha chains, each of them is called as only an alpha chain, and three alpha chains are kept together to form a single helix, a collagen helix. That basically means a triple stranded helix of collagen due to three alpha chains. How many chains? Three alpha chains. Each alpha chain, if you look here, there are some black proteins which are mentioned here, correct? The black proteins. And then you have two other proteins in between. Each of those black protein is made up of glycine, and these other two protein could be X or Y. The x of y here need not be the x of y in this place. In this place could be some other x and y. In this place could be some other x and y. Like that you can have different x and y, but glycine is present between every x and y, which makes glycine how much? 33%, okay, by weight for the collagen. How much? 33%. So glycine is present in 33%. Correct? Simple? Right. So apart from glycine, the other important basically amino acid present in collagen is proline. Proline and hydroxyproline. These two are also present, okay, in good amount, almost accounting for about 100 amino acids, okay, and there are nearly about 3000 amino acids in the collagen, okay. Now these proline and hydroxyproline are very important for the structural integrity of the collagen, okay, right. We will continue. Let me erase this. Okay, good enough. Now we go back to the main portion of the protein, okay, the collagen. Yeah, very important also to remember about this other aspect which he is telling here, that the structural role collagen plays in a particular organ, is dictated by the way the collagen is arranged in the tissue. It's not that the collagen is arranged in all the tissues in the same way. In some tissues, the collagen may be dispersed as a gel. Like for example, inside the eye, you have the vitreous humor. The vitreous humor is nothing but a collagen which is present in the form of a transparent gel. Okay, in the cornea, you see the same type of collagen is present in cornea in a way such that it allows the light to enter in, okay, which allows the transmit to light with minimum of scattering. So again collagen, but the way of arrangement is different, giving it a different function totally. Sometimes it is like a gel, sometimes in tendons, in bones, in ligaments, it is more firmer. It basically here it gives you mechanical stress, taking the mechanical stress and prevent mechanical shear. So therefore here it becomes more opaque and trying to give you more strength, or also basically prevention of basically protecting your body from mechanical stress. So the structure of the collagen, the structure of the collagen, the arrangement of the fibrils basically plays a very important role in its function. So the function is determined by that, right? Simple. So this is basically taken from your Lippincott book. Wonderful book. I think all students, you should read Lippincott, right? Good. We will go to the second page of this book. Let me see where I have put it. I think I put it at the bottom. Yeah, it's right here. Good. So let me move the first page out. Good. And now comes the second page. Now the second page basically you're going to tell about the types of collagen. Now interesting part is, there are so many different types of collagen. How many types are there? Greater than 25 types of collagen are present. There are more than 25 types of collagen which are present. Okay, which is an important part to remember. 25 types of collagen types are present. Okay, the 3 polypeptide alpha chains are held together by interchain between one chain to the other chain. What is the bond that keeps the 2 chains together? Alpha chains. How many alpha chains? 3 chains. Keeping the alpha chains together basically are due to the interchain hydrogen bonds. Okay, and basically each alpha chain could have how many amino acids? 1000 amino acids. Okay, they are combined in various types of tissues, and so therefore, okay, it could be basically different types of alpha chains are present, are combined to form various types of collagen. Like type 1 collagen contains two chains called alpha 1 and one chain called alpha 2. So you have two chains. Totally 3 chains have to be present. So out of that, 2 chains are called alpha 1 and one chain is called alpha 2. Whereas type 2 collagen contains 3 alpha 1 chains. So all chains are very similar. So each of these alpha chain need not be very totally similar. So like example in type 2 collagen, you have alpha 1, alpha 1, alpha 1, 3 alpha 1 bonded together becomes basically a triple helix. In collagen type, that is was in type 2, whereas in type 1 contains of 2 chains of alpha 1 and 1 chain of alpha 2. Alpha 2 is basically a polypeptide, totally slightly a different sequence than that of the alpha 1. That way. Got it? So depending on the type of alpha chains that are there, you could have different combinations possible. Now the different types of collagen depending on their functions. So the function basically determines the type of the collagen itself. Okay. So for example, this is a very nice way. So the way you basically classify and what is it that is very important for you is this part. Okay. Remember the types of collagen as given in this chart. Very, very important. So these are the different things that are very important and you need to know. The one is fibril forming, which means fibers, and out of that you have 1, 2 and 3. 1, 2, 3 are fibril forming. Okay. One mainly in the skin. Okay, of most importance is the skin. Then the bone and tendon. 2 now mainly remember cartilage. 3 means mainly blood vessels. Okay, 1 is skin and bone, 2 is cartilage, 3 is blood vessels. Then you come to network forming, those that form a network. That means one's like a net, obviously giving greater integrity. Okay, 4 is basement membrane. Most of the basement membrane over which some other cells are present is basement membrane. So therefore that is nothing but like a network over which other cells are present. Okay, so therefore the I mainly, okay, consists of type 4 collagen, and also after 4 you don't have to remember 5, 6, directly go to 8. 4 and 8 are network forming. So here again you see corneal and vascular endothelium. Okay, the blood vessel endothelium as well as the endothelial layer of the blood vessels and the cornea. Then fibril associated. What is fibril associated? Either the cartilage and the tendon. Again, ligament. Ligament, if you look here, is mainly type 12 as well as cartilage can be again type 9. Fibril forming and fibril associated. Look at the difference. Fibril, because it forms, is 1, 2, 3. Fibril associated are 9 and 12. 9 and 12. 9 and 12. So you just need to remember 1, 2, 3, 9, 12. 1, 2, 3, 3 into 3, 9. 9 plus 3, 12. So these are all fibril. Whereas 4 and 8, your even numbers are always network forming. Right. Simple way by which, we figured out how to remember these things. Right. Now that that part is over, we go to the basic structure of the collagen, right? Good. Yeah, let's see the structure of the collagen, okay, in little more detail. Okay. So typically, as we said, globular proteins are more, rounded, whereas these are long, they are stretched out, and they are in different layers, and their arrangement is very important, and each of these are held together by your very important part, that is your hydrogen bonds, okay, which is very important. So each is made up of three helix, so that's why you called it a triple helix structure, and all containing an alpha chain, different alpha chains, and so each helix is made up of a alpha chain. What is this each helix actually made up of? Okay, if I were to take a single chain out of this triple helix, okay, the first part is basically having a triple chain, okay. So this is what it means, a triple chain. Now the collagen, when it is present, these triple chain, okay, each consider one chain like this, a triple chain, one alpha helix consists of one chain of polypeptide, like that, alpha chain another, alpha chain another, like plaiting a hair, okay, three plaits. Each single three plait of a hair becomes a collagen molecule. Now you take different plaits, okay, three of you having long hairs, consider different people having different plaiting of hair, triple. Now I cut each of your hair and keep it separate next to each other. One plait of hair here, got it? Then the next plait of hair here. Now in between, you see I have left a gap. Again I place one plait of hair here, the next plait of hair here, one triple helix here, one triple helix, again I have left a gap here. Look at this, I am giving a regular gap between the different helix. In each line I put different plaits of hair, and I leave a small gap in between, and the gap is very uniform. Every time I leave a uniform gap, okay. And so when I do this and create multiple lines, multiple layers, okay, I get your collagen structure. So multiple alpha helix which are kept with next to each other with a regular gap in between, and these basically are your heavy staining regions. The gaps are heavy staining. So the gaps, when you basically take this kind of what is called as a staggered arrangement of the collagen fibers, leads to the bands formation. So wherever the gap is there, there you see the light band, okay, the dark band. Wherever the gap is there, you see the dark band. Where the collagen fibers are there, there you see it as a light band. So collagen fibers are seen as light band. So that is why when you take each particular collagen fibril, okay, fibril means what? One fibril consists of multiple alpha chains which are arranged with a gap in between. So one line I can call it as a fibril, okay. So one line I will call it as a fibril. Multiple fibrils, one fibril, another fibril, one more fibril, multiple fibrils. What did it become? A fiber of collagen. Got it? Okay, so this is the way you can remember the structure of the collagen. Remember, a three plaiting hair kept separate. Okay. Good. Let me know whether you're liking the class. Put a thumbs up. Beautiful. We then go to the next part. Okay, let us look at what is happening between each helix. What is the amino acid sequence here, which is important. Okay, so we talked about the role of different amino acids and we said there is a glycine and then a XY. So look at this. This is how each chain is going to take in. So for a collagen, each alpha helix will have a triplet with the 3 different amino acids with basically glycine repeating. So say for example, that's why you call it XY. These are repeating multiple times. XY could be different amino acids. See here you have glycine followed by leucine and heptadine, then glycine, proline, heptadine, glycine, alanine, something else. Okay, that way. So therefore, this is not heptadine, this is hydroxyproline. Okay, glycine, leucine, hydroxyproline. Glycine, proline, hydroxyproline. Glycine, alanine, hydroxyproline or something else also. Okay, so hydroxyproline and HYL is hydroxylysine. Okay, so these are the 2 other very important proteins which are present in alpha 1 chain. Glycine, hydroxyproline and hydroxylysine. Okay, so different basically amino acids play a very important role, but glycine is present. Every 3rd amino acid is a glycine, so therefore it is present in 33%. Now, though glycine is mainly present continuously, he says that proline facilitates the formation of the helical conformation for each alpha chain because its ring structure causes kinks in the peptide chain. So in the peptide chain, wherever proline is there it will create a kink. Okay, the presence of proline creates a kink because proline by itself, okay, has a ring structure. Okay, correct. Proline is a ring structure. Because proline is a ring structure like tryptophan, proline is a ring structure. Okay, so therefore that creates a kink, whereas the other amino acids, many of them are aliphatic. Okay, done. So therefore the presence of proline dictates the helical conformation of the alpha chain. So because it creates a kink, the chain is now no more straight, it is becoming a helix. So you understand now why it becomes an alpha helix? Because of not presence of glycine but because of proline. Glycine, you know, is the smallest amino acid. Okay, it is smallest, so it will fill in the restricted spaces with 3 chains, and so therefore just basically becomes a part of a repeating residue. Okay, and X is frequently generally proline and Y becomes hydroxyproline or hydroxylysine. Okay, that is why such a chain is called as basically glycine XY. The most important commonest chain is, if they ask you MCQ, remember it is glycine, proline, hydroxyproline. What is it? Glycine, proline, hydroxyproline. Yes, look at this. Glycine, proline, hydroxyproline. This is the commonest chain. Got it? Simple. Of course, you can have others, hydroxyproline, hydroxylysine, which are also present. Okay, and the hydroxylation, like for example, now hydroxyproline and hydroxylysine. Why do you get hydroxy? Means what? Due to after the initial transcription of the amino acid polypeptide chain later on as a post translation. You will study in the synthesis as a part of post translation. There what happens? Hydroxylation takes place. Okay, is a post translational modification. Hydroxylation is a post translation modification. Remember that. Okay, and this post translational modification, okay, leads to the formation of hydroxyproline and hydroxylysine, and so therefore maximizes the formation of interchain hydrogen bonds to stabilize the triple helix structure. So in the presence of this hydroxyproline, there is a maximization of the hydrogen bonds. The hydrogen bonds increase whenever this hydroxy structure is present. So the post translation modification gives it more stronger structure for greater hydrogen bonding. There is also glycolization. The hydroxyl group of the hydroxylysine residues of collagen may be enzymatically glycosylated. Okay, so galactose and glucose are attached to the polypeptide chain by a process of glycosylation. Okay, which is also a post translational modification. Okay, done. Now we straight away go to the the biosynthesis of the chain. Biosynthesis, how is this collagen formed, and then we'll see some important conditions where basically medically their role of collagen is affected. Okay, we'll do that much part today's class, and then we'll do elastin in the next class. Okay, done. The other important point, one small point which is told here in this picture here is, see how proline basically gets hydroxylated, okay, in the pro-alpha chains of collagen by prolyl hydroxylase. Okay, this hydroxylase is cofactor is protected from oxidation, okay, to Fe3 plus by ascorbate. So this part is very important. Where does the role of vitamin C play? Vitamin C deficiency leads to scurvy, bleeding gums. You all read that in 12th. Bleeding gums, okay, and so that is because of scurvy. So here in the presence of vitamin C, what happens? Hydroxylase is an enzyme, and that is protected from oxidation to Fe3 plus by vitamin C. So if basically vitamin C becomes less, what happens? Hydroxylase get converted, becomes oxidized. So hydroxylase will not be present. If hydroxylase is not present, what happens? Proline hydroxylase does not work. If proline hydroxylase does not work, what happens? Hydroxyproline is not formed. If hydroxyproline is not formed, what happens? The hydrogen bonding in the chain, number of hydroxyproline decreases. Hydroxyproline decreases, number of hydrogen bonds decrease. Number of hydrogen bonds decrease, the collagen is weaker. So it leads to loss of cell integrity, and that is why in the basement membrane of the endothelial cells, which is where collagen is very important for stretching and also for helping in clotting in some way, collagen is important, and that is why it leads to bleeding. So you see how important the role of ascorbic acid vitamin C is. How does the role of vitamin C play if they ask you? Simple. Ascorbic acid helps a role by preventing the oxidation, okay, of hydroxylase to Fe3 plus. So therefore presence of ascorbic acid protects the hydroxylase enzyme. Absence of that denatures the hydroxylase enzyme because it gets oxidized. So hydroxylase then becomes deficient, and so therefore leads to absence of hydroxylation of proline to hydroxyproline, and less hydroxyproline leads to less hydrogen bonds and weaker collagen. Good. Now we go to the different synthesis of the collagen. Okay, right. Okay, it's a very elaborate diagram given in this book. I don't think any other book gives you about it. It may not be very, very important, but what is important here is, before you get collagen, every tissue, everything always forms a pro structure. Before collagen, you will get a pro-collagen. Now, what happens here is, you don't only get a pro-collagen, you actually start getting, after transcription, what is known as pre-pro-collagen. That means even before getting pro-collagen, you start getting a pre-pro-collagen. A collagen which is before pro-collagen, and that is a pre-pro-collagen. What are these pre-pro-collagen? They are nothing but your alpha chains. Okay, where is this biosynthesis occurring? These biosynthesis occur at the fibroblast. Okay, where collagen is formed. Collagen is formed in the fibroblast. The same thing if I say fibroblast present in the bone, osteoblast. Fibroblast present in the cartilage, chondroblast. Fibroblast present in the teeth, odontoblast. Okay, simple. So all these three are fibroblasts. The first alpha chain that you get, before you get the alpha chain, you get what is called the pro-alpha chain form, and these are known as the pre-pro-alpha chain. Okay, what do you call them? The pre-pro-alpha chain. This is the first part. Okay, so you have your DNA, you have your mRNA which is lined up here, and then you have your ribosomes on the rough endoplasmic reticulum which are now arranging on it. The mRNA gets formed, and then the transcription is going to take place. So the genes for the pro-alpha 1 and the pro-alpha 2 chains are transcribed into mRNA. So the collagen therefore has got alpha 1 chain and a alpha 2 chain. Okay, type 2 collagen has 3 alpha 1. Type 1 collagen has 2 alpha 1 and 1 alpha 2. You know that. Okay, right. The mRNA is translated on the rough endoplasmic reticulum into pre-pro alpha chain. So once the mRNA gets translated, the translated mRNA itself is called the pre-pro alpha chain. Okay, and these are extruded into the lumen where the N-terminal signal sequence is removed, converting them into pre-pro to pro. So pro-collagen and pre-pro collagen, what is it? Pre-pro collagen is nothing but your mRNA to which the ribosomes are attached. When the N-terminal signal sequence is removed, you call it the pro-collagen. Okay. Once now you have removed the pro-collagen, removed the N-terminal, so therefore what happens? The ribosome is removed now. Okay, the ribosome gets removed. And so before the ribosomes were removed, it was called pre-pro-collagen. Once the ribosome is removed, it is called as pro-collagen. Okay. The next part that is done here is basically wherever proline and lysine is there you hydroxylate them. The next part is hydroxylation. Put one hydroxyl group to wherever proline and lysine is there. So becomes what? So if you look at this, it is hydroxylation therefore is a pre-translation. Okay, whereas post-translation is hydroxylysine or post-translational modification. They are post-translational modification. After the mRNA is formed, then the polypeptide chain. Here you see the polypeptide chain is already lengthened. The mRNA polypeptide chain is already lengthened, okay, which is still attached to the ribosomes. It is attached to the ribosomes, but the N terminal alone is removed. So the ribosome is still attached now. Okay, so but translation has taken place, the polypeptide chain sequence is completely formed, and on that polypeptide chain hydroxylation takes place. Then what happens next step? Wherever selected hydroxylysine is there, the hydroxylysine gets glycosylated. Glucose and galactose get added to it. Then after that what happens? The three chains, now different polypeptide chains are there now. One chain from one mRNA polypeptide chain, another chain from another polypeptide chain on another ribosome. Another ribosome has one more polypeptide chain, alpha chain. So three alpha chains have come together. They start to assemble and form interchain disulfide bonds. The interchain bonds could be hydrogen, we said, very important, and also disulfide bonds are formed, okay, between the chains as well as interchain. You get like between the alpha helix itself, one helix, two helix, between one area, one chain, like how you have intra and inter disulfide bonds in immunoglobulin. Remember that in 12th standard, same way. So inter and intra disulfide bonds which are formed. Okay, and these basically are formed where, which part? In the C terminal, in the C portion, not in the N portion. N terminal is already removed straight away, okay, initially, and that is when the N terminal is removed, it becomes the procollagen. Before the N terminal is removed, is called the pre-procollagen. Now the C terminal area, these disulfide bonds are forming. This will lead to more helical structure, and that moves from the C terminal towards the other side. As it is formed, it will become the triple helix gets formed. Once the triple helix formed, the procollagen molecule is secreted from the Golgi vacuole into the extracellular matrix. The procollagen now has come out of the cell, okay, into the extracellular matrix. This procollagen, then N-terminal and C-terminal propeptides are cleaved by the procollagen peptidase, producing a tropocollagen. Okay, so the N-terminal, so you can get a N-terminal and C-terminal propeptides are cleaved by the procollagen peptidase. Okay, so there is a procollagen peptidase which cleaves them and basically producing the tropocollagen. Okay, I'll just read that part a little more better. After hydroxylation glycosylation, three pro-alpha chains from procollagen, a precursor of collagen that has a central region of the triple helix flanked, okay, the central region of the triple helix is flanked by a non-helical N, correct, one side, see here you have a non-helical, this side is not very helical. There are some disulfide, but it's not too much. So that is called the N-terminal. And the C-terminal, okay, the non-helical, this part, this whole thing becomes helical, whereas one end of it is less helical, and that becomes, which is more pleated. Okay, like your hair, one end of it is not very pleated, right? So that is like the N-terminal. The other end is also the C-terminal, again, which is not very much pleated. So these are the C-terminal extensions of the pro-alpha collagen. This brings three alpha chains into alignment favorable for triple helix. The procollagen moves through the Golgi apparatus where they are packed into secretory vacuoles. The vesicles fuse with the cell membrane, causing the release of the procollagen matrix. Okay, after the release, the triple helical procollagen molecules get cleaved by, now you see this is a triple helix, but this triple helix that is there, correct, has got this very important part. Understand this well. What is the difference? You have this triple helix procollagen. This whole thing structure, what you're seeing here, is your triple helix procollagen. Okay, up to this portion, this whole thing. Okay, let me just minimize that. This whole structure is your procollagen. It is coming out of the Golgi apparatus. Now what is happening? Right at this is the helical structure which is more already bonded. Edge here is unpleated. This is also unpleated. N terminal, C terminal. Remove the C terminal which is not wanted, not well pleated. Remove this N terminal which is not pleated. Now this remaining becomes a tropocollagen. One tropocollagen here, another tropocollagen here, another tropocollagen there. With a gap, these three becomes one fibril. Multiple fibrils then become your fiber of collagen. So the tropocollagen is basically what? Already multiple, where the C terminal, N terminal are removed by the peptidyl, by the terminal peptidases, C and N procollagen peptidases. That is what is removed. This leads to orderly arrangement of the fibril, and that basically becomes what is known as a collagen fibril formation. Okay. Now, there is a quarterly staggered triple helix. Collagen is called a quarterly staggered. What do you mean by quarterly staggered? This word is given very importantly. You call that quarterly staggered. Okay, I'm going to write that. Quarterly staggered triple helix. Okay, so this is very important to remember. Now, why do you call this quarterly staggered? I told you that to form a single fibril, you need multiple plaits. Okay, each plat becomes a tropocollagen after the C terminal, N terminal is removed. Now, the gap that is there between them, okay, the gap that is there between them, this particular gap which is there, this gap is 1/4 the length of each particular tropocollagen. Okay, this is a tropocollagen, this is another tropocollagen put together. If I were to take one single plait of hair, 1/4 the length of the plait of the hair is the gap. What is it? Gap is 1/4 the plait of the hair. That is why you call this quarterly staggered triple helix. Got it? Fantastic. Now comes the last part, the cross-linking formation. What is it? The cross-linking. The fibrillar array of collagen molecules serves as a substrate. Okay, now there are different fibers that are present. Okay, this fibrillar array of collagen molecules serves as a substrate for the main enzyme lysyl oxidase. Remember this enzyme very importantly, lysyl oxidase. This copper containing extracellular enzyme, okay, we'll go and read that. Yeah, correct. We'll just try to read that part. Where did I put that? Yeah, it's there down here. Yeah, cross-link formation. Okay, the fibrillar array of collagen molecules serves as a substrate for lysyl oxidase. This copper containing extracellular enzyme, remember this, it's a copper containing enzyme, lysyl oxidase, okay, and what is the role? It oxidatively, it is an oxidase, so it will use oxygen. Deamines, removes the amine. Some of the lysine and hydroxylysine residues are removed. Deamines. So it removes the amine from the lysine and the hydroxylysine. The reactive aldehydes that result, okay, they are known as allysin and hydroxyallysin. So they become now no more amines. From amines they become aldehydes. So they are known as hydroxylysine, allysin and hydroxyallysin. Can spontaneously condense with lysine and hydroxylysine residues in the neighboring collagen molecules to form a covalent cross-link. So see how cross-linking takes place. The cross-linking doesn't take place from outside to another molecule. It is totally from the adjacent molecules itself. So adjacent strands will start linking with each other and becoming a cross-linking. So this cross-linking is one linking from one to the another between the different fibrils. From one fibril to another fibril, one line to another line. Okay, what happens? There is this lysyl oxidase which basically removes the amine, converts hydroxylysine and lysine to aldehydes, and that then creates, okay, a cross-linking with the neighboring portions which have lysine and hydroxylysine, and so therefore cross-link can form between two allysin residues, and so that cross-linking is very important for the integrity. Like for example, eye has a very important part called the cornea. The cornea consists of collagen. We said that already. Type 3, type 4 collagen, correct, is also present, as well as sometimes type 8. Now, among this cornea collagen which is present, in the presence of deficiency of this lysyl oxidase, which is copper containing, okay, cross-linking will not take place. If the allysin don't form, cross-linking can't take place. So if cross-linking doesn't take place, the strength of the fibers become weak. If strength of collagen fibers in the cornea become weak, it leads to bulging of the cornea, and this problem is known as keratoconus, okay, where cornea becomes more curved. Lysyl oxidase is also one of the very important, okay, copper containing enzymes. Apart from that, there are others like, for example, ceruloplasmin, cytochrome, superoxide dismutase, tyrosinase. Okay, so if copper gets affected, okay, copper hemostasis, disruption in copper hemostasis causes copper deficiency, that is Wilson's disease, Menkes syndrome. These are the conditions where there is a copper deficiency or a overload. Okay, disruption of copper either because of deficiency is X-linked Menkes syndrome, or overload Wilson's disease. In both of this, copper metabolism gets affected. If copper gets affected, lysyl oxidase gets affected, leads to more other problems. Okay. Now we go to a very important part basically, which is the collagenopathy. Okay, the different collagen affecting conditions. Remember two main conditions. In third one, you already read it, that is the scurvy. You know that what happens in scurvy, we talked about it, that scurvy is because of ascorbic acid which is needed for the, which portion? For the prolyl hydroxylase enzyme. So the hydroxylase enzyme doesn't take place, hydroxylation of proline doesn't form, hydrogen bonds become weak, leading to bleeding, bleeding problem and bleeding comes. So that is the role of scurvy. Now we come to the next part, two important conditions. One is known as Ehlers-Danlos syndrome. What is it known as? Ehlers-Danlos syndrome. Okay. These are known as collagenopathies. There are about more than 1000 mutations have been identified in 23 genes coding for 13 of the collagen types. Now, the Ehlers-Danlos syndrome is a kind of a heterogeneous group of collagen disorders that results from hereditary metabolism defects in fibrillar collagen. So, hereditarily, there is a problem as a result of which, for example, same thing. You have two enzymes, lysyl hydroxylase as well as N-procollagen peptidase. So, if there are mutations for any of these enzymes, it will lead to defective type 1 collagen, type 3 collagen and type 5 collagen. Type 1 is typically present where? In the skin. Type 1 typically is present where? In the skin. So, deficiency of that leads to a very elastic skin. It's very loose skin. You need some amount of collagen is meant for maintaining the strength and the sturgidity. So, because it is poorly present, it makes the skin very thin. You can stretch the skin very much like that. Similarly, ligaments can be easily stretched. You have seen lot of people where the ligaments are nicely stretched. They can hyperextend the digits. What can they do? Hyperextend the digits. So, this is known as skin extensibility, fragility, joint hypermobility. The joints and all, you can bend it backwards and all. You've seen a lot of girls, young girls, doing all this kind of yoga stunts with hyperflexible joints. They're all people having this Ehlers-Danlos syndrome. You know, many of them have this, and so they were like hyperextensible. It looks, wow, they are able to do all these things very well. So, because of hyperextensibility of the type 1, type 3, type 5 collagen. The same thing also is present from the blood vessels. Okay, correct. So, 1 is present in the skin mainly, predominantly we said. 3 is present in the blood vessels and skin both. So, 3 is present in the blood vessels. So, therefore, even the blood vessels should get affected, can lead to potential arterial rupture. Dangerous. Sometime Ehlers-Danlos syndrome children, that's why need to be scanned. If you see children with hyperextensible joints or anything, immediately ask them to get their heart scanned, echo done, because they could be having a huge aneurysm of the aorta. They could be having a arterial rupture can happen due to poor blood vessels. Okay. So, see how we are able to nicely link this. Okay. Collagen that contains mutant chains may have altered structure, secretion, distribution and frequently is degraded. Okay. So, incorporation of just one mutant chain may result in degradation. Yeah, even if you have a single mutant ninja, okay, single mutant ninja collagen, then you call it a dominant negative effect. Even a single dominant mutation can lead to a negative effect. The other important condition is known as osteogenesis imperfecta. Simple. Remember, osteogenesis means what? Bones have to be formed. If the formation of the bone is imperfect, you call it a osteogenesis imperfecta or OI. Okay, it's bone fragility. Obviously, because the bones are not well formed, continuously formed, so therefore the bones are very weaker, easily prone for fracture even with minimal trauma or no trauma. It is caused mainly by dominant mutations to the genes that encode the alpha 1 and the alpha 2 chains of the collagen. So, osteogenesis imperfecta is formed due to basically poor formation of the alpha 1 chains of the collagen in the bones, okay, due to the dominant mutation. Again, what type? Due to dominant mutations. And these cause, what happens? The replacement of glycine by amino acids with bulky side chain. Now, instead of putting glycine, proline, hydrolysine, you start putting glycine XY, you know that. Instead of that, you replace glycine with something else, with bulkier amino acid. Now you put a bulkier amino acid instead of glycine. So what happens? The typical ratio is lost. So therefore, the bulkier chain is easily prone to be broken. Okay, and so that leads to bone fragility, intrauterine fractures. You can get a fracture inside the uterus in the stillborn itself. See this picture here, you can see very nicely that the child is actually almost born, stillborn, but there are multiple fractures here, here. Okay, so multiple fractures which are present due to the osteogenesis imperfecta. It can also lead to bone fragility, hearing loss, and also blue sclera. We said that, okay, blue sclera, because why? Simple. Blue sclera happens because the sclera is very thin. Then it shows up the inner blood circulation. Inside of the sclera, you have what is called as the choroid. The choroid is the blood supply of the eyeball of the back. So the choroid consists of a lot of veins. So the veins start appearing more prominent if the sclera is thin. Wherever you would have seen some people have a little bluish eyes, not the cornea. I'm telling about around the white, the sclera. So that portion, if it appears blue, it is because of a thin sclera. Okay, they are short stature, spinal curvature is humped back, they have a kyphotic appearance, okay, blue sclera. And this typically is due to because of either type 1 or type 3. Okay, type 1 is most common, characterized by bone fragility, hearing loss, blue sclera. Type 2 is most severe, typically lethal. So there are 3 types. You will learn this more in medicine, but good to remember that there is one type which is lethal, and that is type 2. Okay, and many of them die due to pulmonary complications. And type 3, so the type 2 is the most lethal form. And type 3 can have basically spinal curvature, kyphotic appearance, blue sclera, and also poor dental development. So fantastic. With this, we cover most of your collagen. I think you all will like this portion. Please go through it and ensure that you score full marks on collagen. Remember to see the structure of the collagen. Don't forget to read Lippincott. It's an amazing book. You will not forget what you have read. Perfect. Thank you.